Good day, the great turns of life sciences. My name is Mr. Motsei Ramukunupi, and today we will be looking at revision and some of the topics that have been covered since the reopening of schools. So today, basically, we will be looking at some of the topics that were covered in Term 1, and then during the pandemic, we know that we have lost so much time, contact time, that we were not able to cover some of the work. As you are familiar that some of the topics in our curriculum have been removed and some of the topics were reduced. So basically I'll be taking you to this document over here, which is the Free State Department of Education Annual Teaching Plan for Life Sciences. Well, it is very important that you access this documentation, as it clearly indicates which topics are covered and which topics have been removed from the curriculum for this year, since we experienced the pandemic and we did not have enough time to cover some of the topics. We'll just kick in with today's session. We'll be looking at the chemistry of life. In the chemistry of life now, we'll be looking at the inorganic compounds. Now we know that inorganic compounds are compounds that don't mainly have carbon, hydrogen, and oxygen all together. For example, we have sodium, potassium, calcium, phosphorus, iron, and iodine. And these are basically the macronutrients that we require in large quantities in our body. And we're also going to look at organic compounds such as sugars, carbohydrates. Sugars can exist as single sugars or as single units of sugar, such as glucose and fructose. And sugars can exist also as disaccharide, where we have two molecules of sugars joined together, for example, sucrose and maltose. Or they can be a chain of single units of monosaccharides that can give us polysaccharides, for example, starch, cellulose that we find in plant material, and also we know that the plant cell is composed mainly of cellulose, and another example of a polysaccharide is glycogen. We're also going to be looking at lipids, which is an example of organic compounds, proteins, and we know that the characteristics of proteins is that they are sensitive to temperature and pH. Proteins, what happens in high temperatures or at high pH is that they lose their structure and their functioning, or in very low temperatures they become inactive. We'll be also looking at the lock and key model of how enzymes work and enzymes in everyday's lives. Also looking at nucleic acids, DNA and RNA, which consists mainly of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And we'll be looking at vitamins. We'll be also looking at an overview of microscopes, different parts of or different organelles within the cell, and their roles in the cell, or what are the functions of different organelles in the cell. As we know that cells, they differ in size and shape and structure. Basically now we'll be looking at differences between plant and animal cells. We'll be looking or tabulating the differences between the two. Organic molecules are made up of carbon and hydrogen and oxygen, while some other they contain elements such as nitrogen and phosphorus. Now the cells are made up of proteins, carbohydrates, lipids, nucleic acid, and vitamins. Now we know that proteins, carbohydrates, lipids, nucleic acids, and vitamins, they are organic molecules, as they contain carbon, hydrogen, and phosphorus, and some of other elements as well. Now carbohydrates, they contain carbon, hydrogen, and oxygen. Now monosaccharides are single sugar units. They only have one unit of sugar. For example, we have glucose and fructose. Now, when we join two monosaccharides together, then we form a disaccharide. This will be double sugars. Example of disaccharides, we have maltose and sucrose. Polysaccharides are many sugar units. Now, if you take many monosaccharides, for example, if you take many glucose molecules and you join them together, it will form polysaccharides such as starch. And other examples of polysaccharides include glycogen and cellulose. Now, the biological importance of carbohydrates is that they are the source of energy. They provide us with energy, as we know that carbohydrates are sugars, and sugar gives us energy. Storage of energy. Glucose is stored as starch in plants, and then they are stored as glycogen in animals. Now, this serves as energy reserves. We know that maybe when sugar levels are low in the body, glycogen that was stored in the body will be broken down into glucose so that sugar levels in the body will be restored. And also they serve as structural function. We know that the plant cells of the walls of plant cells mainly are made up of cellulose. Lipids are example of organic compounds which contain carbon, hydrogen, and oxygen. It includes fats, waxes, and oils. Now, one molecule of lipid consists of a fat molecule, consists of one molecule of glycerol and 3 molecules of fatty acids. Now, this means that a lipid is made up of glycerol and fatty acids. Now, we have unsaturated fats. Now, unsaturated fats are a type of fats that are liquids at room temperature. They mainly include plant fats such as olive oil and sunflower oil. And then saturated fats, these are type of fats that are solids at room temperature, mainly animal fats such as red meat and butter. We know that they are solids at room temperature, so therefore they are saturated fats. Now, a diet high in saturated fats can cause problems in the body, such as cholesterol, and it can lead to other complicated conditions where now these fats will clog the arteries and hinder with the blood flow to major organs in the body from the heart, and this can lead to heart attack or atherosclerosis. Now, the biological importance of lipids is that they are a rich source of energy, storage of reserve energy. They also serve as structural component in cell membranes. They provide insulation beneath the skin. They warm up the skin, and also they protect major organs, including the heart and the kidneys. Proteins. They contain carbon, hydrogen, oxygen, and nitrogen, and also sulfur and phosphorus. They consist of long chain of amino acids. We know that amino acids are monomers of proteins or the building blocks of proteins. The structure of protein is disrupted by extreme temperature and pH, and this is denaturation. When proteins are exposed to high temperatures and pH, they lose their structure and their functioning, and that is termed denaturation. Biological importance of proteins: 50% of dry mass of living organisms. As enzymes and as hormones, as enzymes, we know that proteins, they are biological catalysts, and as hormones, they are chemical messengers that send messages to target organs. And we know that all enzymes and all hormones are made up of proteins. They also serve a function in transport, as hemoglobin transports oxygen in the blood. They also protect the body from infections, as antibodies, they are proteins, fibrinogen, and also in blood clotting. enzymes, as we have mentioned previously, that all enzymes, they made up of proteins, but not all proteins are enzymes. Now, enzymes, they are biological catalysts, which means they speed up reactions in the body. Now, without enzymes, now these reactions will occur very slowly in the body. Now, if you look at the lock and key models of enzymes, the behavior of enzymes or how they work. Now this will be an example of a lock and key model where we have an enzyme. Now we see that now the active site of an enzyme will allow a substrate to actually fit in with the enzyme. Now we have the substrate, we have the enzyme, and then the enzyme have an active site where now the substrate will be binded with the enzyme. Now when they have binded, now we have the enzyme substrate complex, and then they will react, and then at the end we will have products that are formed. But as you notice, the structure of the enzymes remain the same, while the products now have been formed and now they are different from the substrates. While other enzymes, they act as builders, or they separate molecules. They act as catabolic agents, or they build molecules. They are anabolic agents. Now, enzymes, they work at optimum levels. They work at levels which they act best. Now, as I've mentioned, that enzymes, they are sensitive to temperature and pH. They function best at optimum rate. Depending on type of enzyme that is acting, now it will require a certain different type of optimum rate from another enzyme, as they are different. Now, if we look at this type of graph where the effect of temperature on enzyme activity, we see now when temperature increases, we see the enzyme activity also increases up until it reaches the peak. Now at this peak, this is the optimum temperature of this enzyme. Now we can determine the optimum temperature of this enzyme, saying that the optimum temperature of this enzyme, based on this graph, will be between 38 and 39 degrees Celsius. This will be the temperature at which this enzyme works best. And beyond the temperature, as we increase the temperature from 40 degrees Celsius, 50, 60, 70 degrees Celsius, we see now the enzyme activity decreases. Now this is because now at extreme temperatures or at very high temperatures, the enzymes, they lose their structure and their functioning. We say now that they denature and lose their function, and now this is why now the enzyme activity starts to decrease beyond the optimum temperature. In another graph here, we have different types of enzymes: pepsin, sucrase, and amylase. Now we see now they have different optimum pH levels, as these enzymes mainly they work in different organs of the body. So for example, amylase we'll find in the saliva, and then optimum temperature of amylase will be 7. This is the optimum pH level of this enzyme. This will be the optimum pH at which this enzyme acts best. This will be different from pepsin, where now we see the peak on the graph there is 2, the pH of 2, which means pepsin functions best at pH of 2. And then sucrase functions best at a pH of 5. Now, what happens now when you increase the pH levels beyond 2 for pepsin? We start seeing now the enzyme activity for pepsin starts to decrease. This is because now pepsin has been exposed to pH levels beyond the optimum pH levels. Now pepsin will start losing its structure and functioning, and then the enzyme activity will start to decrease. Also for sucrase, we know now it's indicated here that sucrase functions best at pH of 5, and beyond that we see now the enzyme activity of sucrase decreasing. Also for amylase, amylase functions best at the pH of 7, and beyond the pH of 7 the enzyme activity of amylase starts to decrease, because now the enzyme has been exposed to high pH levels, and then now they start losing their structure and their functioning. Vitamins consist of carbon, hydrogen, oxygen and nitrogen, and also sulfur and phosphorus. Now vitamins are required in small quantities for normal growth and development in animals. We have different types of vitamins. We have vitamin A, where we can get vitamin A, the food source. We get it from carrots, from dairy products, from fish. And vitamin A is very important in good eyesight and healthy skin. When you don't get enough vitamin A, the deficiency disease that can develop is poor night vision and a dry, hard skin. Vitamin B1, we can get this type of vitamin B1 from whole grain cereals, from lean meat and from liver. And now it serves a purpose for healthy appetite and prevents constipation. Now the deficiency disease, when now you don't get enough vitamin B1, is beriberi, nerve and heart disorders. Vitamin C, which we get from citrus fruit such as lemon, oranges, tomato, and we also get it from green vegetables. It functions to prevent infections, maintains blood vessels, walls, in the gums and our teeth. Deficiency disorder, if you don't get enough vitamin C, is scurvy and the bleeding of gums and underskin. Vitamin D, which we can get from dairy products, fish, liver oil, and from the natural source, which is the sunlight, which functions for absorption of calcium and building of strong bones. Now the deficiency disease, if you don't get enough vitamin D, the bones in children, in the lower limbs, they start to bend. This leads to rickets in children and osteomalacia in adults. Vitamin E, which we can get from meat, dairy products, and wheat germ, functions for vitamin E is that they build red blood cells, they protect the body against cancer and heart diseases. It can lead to infertility and anemia if we don't get enough vitamin E. Nucleic acids, we have two types of nucleic acids that we find in the nucleus of the cell. And this is DNA, which carries hereditary information, the deoxyribonucleic acids. And then we also have RNA, which is the ribonucleic acid, which is very important in protein synthesis. Now, these two types of nucleic acids are built from monomers that we call nucleotides. So nucleotides are the building blocks of DNA and RNA. Now, the biological importance of nucleic acid is that the DNA carries and transmits genetic information. DNA and RNA control metabolic activities of the cell, such as protein synthesis. Now we're looking at electron microscope. We know that microscopes are devices, or is a device that we require to look at specimens that are very small to be noted with a naked eye or with a normal human eye. So some of the organisms, such as bacteria, fungi, or the cells, that are very small so that we cannot be able to see them with a naked eye. So we require a device an electron microscope, which consists of an ocular lens or the eyepiece, which magnifies the size of a specimen, a body tube. This is the arm of the microscope, which we use to lift up the microscope or to carry the microscope. And we have the revolving nose piece, which have objectives attached to them. Now we have different magnification sizes of the objectives. We can get 4 times, 10 times, 40 times magnification, even 100 times magnification. So now the main purpose of the objective is also to magnify the size of the specimen. So together with the ocular lens, the objective, they magnify the size of the specimen. We have the stage where now you can put the slide of the specimen that will be viewed using an electron microscope, and now we have the stage clips that will hold the specimen on the stage, prevents it from moving, or the glass slide from moving, so that the specimen can be viewed using the microscope. We have the diaphragm, which can be rotated to control the amount of light that comes from the light source. If it is too bright, then you can adjust the diaphragm so that it dims the light, or if it is too dim, you can adjust the diaphragm so that it can become brighter. Now we have the coarse adjustment knob, which we use to move the stage up and down in order to bring a fine focus on the specimen so that the specimen can be viewed very clearly. We also have the fine adjustment knob. Fine adjustment knob slightly moves the stage so that the specimen can be focused and image can be clearly viewed. And we have the base of the microscope. Another type of microscope, very big microscope, mainly used in research in universities, is an electron microscope, which now these are used to view very, very small objects like viruses. They can be used or observed using an electron microscope. Now it is very important so that you note how to calculate maybe the size of the specimen or magnification size of the specimen, or different calculations that are done when using a microscope. For example, here we have a formula that we use to calculate the total magnification. We know that the ocular lens or the eyepiece, they magnify the specimen that is viewed under microscope, and also the objective also magnifies the size of the specimen that is viewed. So the formula that we use to determine the total magnification is that we take the eyepiece and then multiply by the power of the objective. Now let's look at an example 1.1. Using the information given in the diagram above, what will be the total magnification of the specimen be? Show all your workings. Now if the power of the eyepiece is 20 and the power of the objective is 40, we say 20 multiplied by 40, and we say now the total magnification is 800 times. This is not x, but this x means it has been magnified 800 times. Let's look at another object example 1.2. What is the total magnification of light microscope if a 10 times ocular and a 40 times high power objective was used to observe a specimen? Now we have already been given the magnification size of eyepiece or ocular lens, and we are given the magnification size of the objective. We know that our formula that we use there is that the total magnification will be equal to the power of the eyepiece multiplied by the power of the objective lens. Now it will be 10 multiplied by 40, which now the total magnification of the object has been magnified 400 times. Let's look at another example, example 1.3. You are viewing a plant cell under a compound light microscope. You are using a 10 times eyepiece lens and a 40 times objective lens. Calculate the total magnification with which you are viewing the cells. We are already given the magnification size of the ocular lens or the eyepiece, and we are given the magnification size of the objective. We multiply the two, and then we can determine the total magnification, which now is 400 times. One of the most important things when calculating maybe the size of the specimen or the total magnification is the conversion of units. Now, units can be given as millimeters, and they get given as micrometers, or they can be given as nanometers. Now, to convert a millimeter to a micrometer, we just multiply by 1000. And then to convert a micrometer to a nanometer, we multiply again by 1000. So for example, if the size of the specimen is 4 millimeters, but we convert it to micrometers, we multiply by 1000. And then to convert it to nanometers, we also multiply it by another 1000. But what do we do if we need to convert from nanometers to micrometers to millimeters? From there, we divide by 1000. So if we convert 4 million nanometers, divide by 1000, it will give us 4000 micrometers. And then if we convert it to millimeters, we divide by 1000, and this will be equal to 4. Now, when given calculations of different units, it is very important that firstly you convert the units to the same units before you do the calculations. Now, let's look at other examples that involves conversion of units. Now, the formula we use for magnification and actual size, we say now magnification will be equal to the measured length of the scale bar and the scale bar, and now we can determine the magnification size. Remember, now we said the units are very important, the units needs to be the same. All measurements must be in the same units. This is very important. Now, if we determine the actual size using the scale bar, we'll be using the formula saying magnification, the size on the diagram, the number of the scale bar, measured length on the scale bar. Now you'll be requiring a ruler there to determine that, and then must make sure that all these units are the same. And when we determine the actual size without the scale bar, said now the actual size is the measured length divided by the magnification. We can look at other examples. Here we have examples of different types of, it's not even different types, but we're looking at the same type of cells, but in different magnification sizes. Now, if you note here, we have A, B, and C. Now, where C is viewed in a larger magnification as compared to B, and now you can see A, the magnification size is very small as compared to B and C. Now, we have the following microscopic views represent magnification of 100, of 1000 times, and 40 times. Now, we need to determine now which one was viewed under 40 times now will be A was viewed under 40 times magnification, and followed by B, which was viewed under 100 times magnification. And we see now the structure C there is even larger than B and A. So this one we can determine and say that it was viewed under 1000 times magnification. Example 4.2 here, we need to calculate the magnification for this scale bar by showing the working. Now this will differ in different sizes of the page. Now maybe an A4 will differ from an A5. If we need to determine the total magnification of now we use the formula saying the measured length of the scale bar. Now how do we determine that? The measured length of the scale bar, you take your ruler and then you measure there in millimeters. It gives us 30 millimeters. So the measured scale there is 30 millimeters. But if you look at the scale bar itself, the units there that are used is in nanometers. So we need to make sure that the units are the same. Either we use nanometers or we use millimeters. So now we convert the millimeters to nanometers or convert the nanometers to millimeters. So the measured length of the scale bar we have measured using a ruler is 30 millimeters. Now we divide it by the scale bar that was given, which is 100. Now to convert from millimeters to nanometers, we know we multiply by 1000. So it will be 1000 now, and then it gives us micrometers, and then we multiply by 1000 again, and this will give us the value in units of nanometers, and then divided by 100 nanometers, and then our answer there will be the total magnification is 300,000 times. Or if we change the units to millimeters, we will divide the nanometers by 1000 two times, and then the answer there will give us 0.000003 millimeters. And we have already determined the length of the scale bar, which is 30 millimeters. It gives us the same number as the total magnification of 300,000 times. So make sure that you know how to calculate the microscope calculations, determining the total magnification of an object or determining the size of the specimen. Now moving on, we'll be looking at plant cells and the parts that or organelles of the cells that we find in plant cells. Now in plant cells, we know that the shape is fixed, it does not change. It is made up of an exterior cell wall which provides shape and strength and also protection of the internal organelles of the cell. Now a plant cell has a chloroplast, which is very important in protein synthesis, where now plants make their own food using light. This is a difference now when it comes to animal cells, as animal cells do not have chloroplast, and animal cells also do not have a cell wall. Look at ribosomes, which are very important in making of protein or protein synthesis. And now the large vacuoles for the storage of water, metabolic waste products, and pigments. Now that is another difference between plant and animal cells. As animal cells, they have no vacuole or very small vacuoles. As plant cells, they have a large vacuole for the storage of water and metabolic waste products and pigments. We have the smooth endoplasmic reticulum, which is very important in the transportation of proteins. And we have the rough endoplasmic reticulum, which collects and transports proteins. Now, the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum is that the rough ones, you can see we have ribosomes that are attached to the endoplasmic reticulum. We have the nucleus of the cell, which consists of a nuclear membrane, which forms the nucleus and protects the DNA. We have the nucleolus, which is the region where DNA makes RNA for protein manufacture. We have the nucleopore. This is the movement of substances in and out of the nucleus. We know that now when maybe proteins are made, the mRNA will leave the nucleus to go into the cytoplasm of the cell. We have the mitochondrion of the cell, which is very important in the production of energy through cellular respiration. And we have the cell membrane of the cell, which is very important in controlling what substances or which substances enters or leaves the cell. An animal cell, which is different from the plant cell, we can see now, animal cell does not have a cell wall surrounding it. We only have the cell membrane of the plasmalemma, which controls the entry or which substances will enter or leave the cell. We have Golgi apparatus, which collects and transports proteins. The mitochondrion of a cell, which produces energy through cellular respiration. Now, if you can see here, the vacuole of animal cell is different from the plant cell. The vacuole of a plant cell is large, and the vacuole in animal cell is very small. So it is very important to know different parts of the plant and animal cell, and also be able to state differences between the plant and animal cells. Now, in animal cell, when we look at the shape, is that it is irregular. It is not fixed, keeps on changing. But in plant cell, the shape of the plant cell is fixed. Animal cells do not have cell wall, while the plant cells, they have a cell wall. Animal cells don't have chloroplast, but we find that chloroplast is present in plant cells. And then the vacuole in animal cells is small or sometimes absent, but in plant cells there is a large vacuole that is present. We're just gonna break for 5 minutes, and then when we come back, we look at cell division that is termed mitosis. When we come back, we're looking at mitosis, the cell division, and now we'll just break for 5 minutes. Welcome back from the short break. Now we'll continue with mitosis. Now in mitosis, when we define mitosis, is a cell division where a cell that divides, termed the mother cell, will produce two identical daughter cells with the same number of chromosomes as the mother cell. Now this basically means that when one cell undergoes division, it will split up into two and then forms two identical cells. But these cells that are formed, which are daughter cells, will have the exact number of chromosomes as the initial cell, as the mother cell. Now, if maybe we draw a diagram of a simple cell there, and then if, for example, this cell has 46 chromosomes inside the nucleus of a cell, if the very same cell undergoes mitosis to form two identical daughter cells. Now, instead of the cells having 23, mitosis ensures that the two cells that are formed, they each have the same number of chromosomes as the mother cell. Now, before a cell undergoes a cell division, it goes through a phase of interphase where now the chromatic network inside the nucleus of the cell will double. Now interphase happens before a cell can divide. Now in mitosis, chromosomes is a structure found in the nucleus of the cell made up of a centromere that joins two sister chromatids. Now if we do a structure of a chromosome, or if we draw a chromosome, it is made up of two sister chromatids which are joined together by a centromere. So this will be sister chromatids joined together by a centromere. So the functioning of the centromere is to join two sister chromatids together. Mitosis is divided into 4 different phases, which include prophase, metaphase, anaphase, and telophase. In interphase, we know that interphase occurs before a cell can undergo a cell division. Now we know that during interphase, the DNA in the chromatin network will duplicate, become double, and the DNA thickens and then becomes visible. In prophase, each chromosome is visible as 2 chromatids joined together by a centromere. The centrosome is made, and then the 2 separate centrioles, and then the centrosomes, they move to the opposite sides of the cell pole, and then the spindle fibers, they form between the centrosomes of the spindle fibers. Now if, for example, we draw just a simple structure of what happens in prophase, we have the cell membrane there, and now the nuclear membrane starts disappearing in prophase. For example, now if we have a cell with maybe 4 chromosomes there, now these chromosomes, they become visible during prophase, and then what we have there is that the centrosomes, they start appearing and forming the spindle fibers in the opposite poles of the cell. So if we label the parts that are involved in prophase, we have the cell membrane, the centrosomes in opposite poles of the cell, and then we have chromosome. This is what actually happens in prophase. Now the nuclear membrane will start to disappear, and then the chromosomes, they become visible during prophase. In metaphase, now the nuclear membrane is completely disintegrated, and now the chromosomes, they line up at the equator of the cell. Now each chromosomes become attached to its centromere, to a separate spindle fiber, and starts to move towards the equator of the spindle or of the cell. Now if we draw or sketch metaphase, we know now in metaphase the nuclear membrane has completely disintegrated and the chromosomes, they lie at the equator or at the center of the cell. Now each chromosome, the spindle fiber attaches to the centromere, which joins the two sister chromatids. We have spindle fibers attached to each centromere of the chromosome. Now this is the events that occur during metaphase of mitosis. In metaphase, the chromosomes will lie at the equator of the cell, and then the spindle fibers will be attached to each centromere of the chromosome that joins two sister chromatids together. And now we see now there is no nuclear membrane that is visible. Now we know that during interphase now the nuclear membrane completely disintegrates. Anaphase is the next phase after metaphase. Now in anaphase, each chromosome separates to its sister chromatids by the action of spindle fibers, pulling each sister chromatids towards the pole of the cell. Each chromatid now is called a daughter chromosome. We then pull to the opposite side of the cell. Now if we sketch the events that occurs during anaphase, now what happens in anaphase is now that the spindle fibers will start to contract and pull each chromatid towards the pole of the cell. Oh, the cell had 4 chromosomes. Now we see now the spindle fibers in anaphase will contract and pull the chromatids towards the opposite poles of the cell. Now we still have our centrosome there. We still have the spindle fibers. And now we have the chromatids that were separated now from the sister chromatid from the chromosome. Now these are separated. Now we have chromatids. Now the last stage of mitosis, we have telophase. In telophase, cytokinesis, which is the splitting up of the cytoplasm. Now the cytokinesis starts by the cell membrane and then starting to constrict at the equator of the cell. The nuclear membrane and the nucleolus, they form in each daughter cell, and each daughter cell has the same number of chromosomes as the mother cell. So basically, during telophase, we see the splitting of telophase, the splitting of the cytoplasm there, forming two identical daughter cells. And now what is important there is that the nuclear membrane starts reappearing, and now the two cells will have the same number of chromosomes as the mother cell. Mother cell had 4 chromosomes before cell division. Even the two daughter cells that are formed after cell division have the same number of chromosomes as the mother cell. Now let's look at just a short video that talks about mitosis.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into 2 identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has 2 sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into 2 sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into 2 daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in 2 daughter cells. In this module, you have learned Mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Now, the role of mitosis, or the importance or the significance of mitosis. Now, mitosis has three important purposes or significance. Mitosis is very important in growth, as multicellular organisms need cell divisions to grow. They all start as a single cell and soon have a huge number of cells. This is because of mitosis. Mitosis is also important in repair, as organisms constantly repair and renew themselves. Worn out or dead cells are replaced through cell division. And very important in reproduction, single cell organisms such as bacteria and protists also reproduce by cell division, binary division, and budding. So they require mitosis for reproduction. Now, when we look at tissues, say tissues are a group of similar cells that are adapted to perform a particular function. Now, when we look at animal tissues, we have embryonic tissues, which are either stem cells. Now, stem cells, we know that they do not have a specific structure or a function. They are important in growth and repairing. And animal tissues, we have permanent tissues. These are the type of tissues that have a specific structure and functioning. Now, as permanent tissues, they can be simple tissues or complex ones, such as connective or nerve tissues, which are important in communication and control. Now, we have three different types of nerves. We have unipolar nerves, bipolar, and multipolar, also known as motor, sensory, and interneurons or nerves. And then we have connective tissues, which are important in the shape, structure, protection, storage, and attachment. Examples, we have adipose tissue, areolar tissue, fibrous tissue, cartilage, bone tissue, and blood tissue. In simple tissues, we have ones that are important in movement or muscles. We have smooth muscles, skeletal muscles, tissues, and cardiac tissues. It also includes the epithelium, which are important in protection, absorption, and gaseous exchange. We have the squamous epithelium tissue, columnar epithelium tissue, the stratified epithelium tissue, pseudostratified epithelium tissue, cuboidal and ciliated epithelium tissues. When you look at the human skeleton, we know that the human skeleton is a collection of bones, and humans as adults, they have 206 bones. Now, the human skeleton is divided into axial and appendicular skeleton. The axial skeleton consists of the skull, which protects the brain, and also consists of the ribs and sternum, which protects the heart and the liver, and then the vertebral column, which protects the spinal cord. The appendicular skeleton, it is made up of the pelvic girdle for the attachment of legs and lower limbs, pectoral girdle for the attachment of arms, upper limbs. Now, if you look at axial skeleton, the axial skeleton includes the bones that form the skull, the vertebral column, the ossicles. These are the smallest bones that we find in the body, and then we find them in the ear, which assist in hearing, the ribs, and the sternum. If you look at the structure of the axial skeleton, it consists of the skull, the ossicles in the inner ear or the middle ear, and then consists of the rib cage and the vertebral column. The appendicular skeleton. The appendicular skeleton includes the bones of the shoulder girdle, the upper limbs, the pelvic girdle, and the lower limbs. When we look at the appendicular skeleton, is the shoulder girdle, consists of the clavicles, scapula, humerus, radius and ulna, and other various bones we find on the hands, the pelvic girdle, the femur, long bone, and then the bones that we find on our foot or on our feet. Now, various functions of the human skeleton. The general functions of human skeletons are movement and locomotions, joints and levers with muscles, protection. Vital organs such as the brain, heart, lungs, and the liver, they are protected by the rib cage. Support, strength, and shape, muscles and internal organs. Storage of minerals. You know that our bones can store calcium, magnesium, and phosphate. And also our skeleton, our bones are important in hearing. We have the three ossicles, which are the smallest bones in the ear, which assist in hearing. And then the formation of blood cells, and then red and white blood corpuscles are formed in the bone marrow. Now this brings us to the end of this notes and slides, and after 5 minutes of break we'll look at some of the possible exam questions or test questions in relation to what we have discussed. So after 5 minutes now we'll be looking at some of the questions and how we go about in answering those questions. We'll just break for 5 minutes and then we'll answer the questions afterwards. Welcome back from the short break. Now we'll begin looking at possible test and exam questions. We'll start with the multiple choice questions. Here, question 1.1. In question 1.1 there we have a diagram that represents osmosis. We know that osmosis is diffusion that occurs in the presence of a semi-impermeable membrane. Now here we have solid molecules. We see now there are very few solid molecules, and water, there is plenty of water as compared to B. Now we expect these molecules to move from where there are many to where there are less up until they are equal through a semi-permeable membrane. So the question says, based on the figure below, which one of the following statement is not correct? Is not correct. Now the movement of solvent molecules will take place from B to A, or movement of solute will take place from A to B. Now we know we can see that in A there is less solute, so we expect more solute to move from B to A. So now the incorrect statement there is B, and correct answer there will be B. Chlorosis. Now, if you note, chlorosis comes from the word chloroplast. Now, we expect it to take place in plants and not in animals. So, it's a deficiency disease that takes place in green plants. So, our answer there will be A. A runner is competing in a 100-kilometer race. Just before completing the race, he is out of breath. He needs energy to finish the race. Which of the cell organelles in the body are most affected by the lack of energy? The answer there is C. We know that mitochondria is the organelle of the cell that is responsible for producing energy through cellular respiration. If you look at question 1.1.4, a carbohydrate that forms the main component of a plant cell, now we know that proteins are not carbohydrates. Starch is incorrect. The correct answer there is cellulose, which forms the main component of the cell wall in plants. Which of the following sets of features are common in both plant and animal cells? A says cell membrane, cell wall. Animal cells do not have a cell wall. B, cytoplasm, nucleus, and cell membrane. C, chloroplast. Know that animal cells do not have chloroplast. And then D also is incorrect. Animal cells do not have chloroplast. So our correct answer there will be B. The next question talks about food tests. The table below shows observation made during tests carried out on samples of a particular food. Malon's reagent, we know that it is used to test for presence of protein, and color observation for a positive test is purple. And then iodine solution test, we use it for to test for the presence of starch in a sample, and a positive test includes starch turning blue-black color, and if it turns brown, it means absence of starch. But then Benedict's or Fehling solution test for glucose, it forms a positive test is observed by an orange-red precipitate that forms. So now this will indicate that the sample does include glucose. So now the correct answer there will be that the food sample does not contain proteins, does not contain starch, but it does contain glucose. So the correct answer there will be B. Diffusion is the movement of molecules from a region of higher concentration to a lower concentration. This is applicable to gases, to liquids, and to solids. So the correct answer there is D. Which of the differences between plant and animal cells is correct? A plant cell contains no chloroplast. An animal cell contains chloroplast. This is incorrect. And contains a large vacuole. Contains a small vacuole. This is correct. Contains no cell membrane. It's incorrect. Plant cells do have a cell membrane. Contains no cell wall. Plant cells do have a cell wall. So the correct answer there will be B. Now the next question, 1.1.9, talks about preparation of a wet mount. Now preparing a specimen to be viewed under microscope. Now organize the diagram in order of preparing a wet mount. The first step will be 4, where we have your last slide, and then using a dissecting needle, you place your specimen on the slide, and then put iodine solution and cover it with a cover slip. So it's Roman figure 4, Roman figure 1, 2, and 3. So our answer there will be D. In question 1.1.10, the total magnification of an animal cell was 400 times. Now, we know that when we determine the total magnification will be equal to the power of the ocular lens or the eyepiece multiplied by the power of the objective. So the total magnification there is given as 400 times. Now we need to, and then what is the magnification size of the objective? We don't know that one. And then we are given the lens magnification of 5. So what is power of the objective? So we will divide by 5, and then 400 divided by 5 will give us 80. So the correct answer there will be 80 times. Which of the following parts of the microscope and their function is correct? Diaphragm concentrate lights, the lamp warps the specimen, the nose piece holds the objective lenses, and the cos focus adjustment find cos. Nope. We know that the diaphragm does not concentrate light but regulates the amount of light that goes to the specimen from the light source, and the lamp produces light, does not warm the specimen. We know that this nose piece, they hold different objective lenses with different magnification powers. So C is the correct answer there. The basic unit of life is, this one is a simple one. We know the basic unit of life is a cell. A disaccharide that is formed when two glucose molecules are combined chemically. Here we know that when you join two monosaccharides together, it forms a disaccharide. So we know cellulose is a polysaccharide, and then sucrose, fructose, and maltose. We know that fructose is a monosaccharide, and then sucrose is a polysaccharide. Which comparison between glucose and protein is inaccurate? Now this goes back to food tests again. Glucose, Nylon's test, we know that to test for presence of is inaccurate. And now we know that glucose is a single molecule, is correct, and then protein consists of many amino acid molecules, is correct. Glucose consists of carbon, hydrogen, and oxygen. So now to test for presence of glucose, we do use valence in A and B solutions, and positive tests give us an orange color, and then if we use valence A and B, and if there is no glucose, it will turn blue. So an inaccurate statement here between glucose and protein will be option A. Because now in Mylon's reagent, when we use this one, the protein positive test will produce a purple color. Let's look at biological terms, question 1.2.1. Organelle responsible for protein synthesis. This will be ribosome. An element that distinguishes proteins and fats. This will be presence of nitrogen. And then the organic substance which contains hydrogen and oxygen in the ratio of 2:1. This will be water. The process that occurs when water body collects farm fertilizers nutrients that increase in concentration that will lead to the decrease in oxygen levels. This is termed eutrophication. A deficiency disease that develops due to a shortage of vitamin D. We know that in children it can cause rickets, or it can cause osteoporosis in adults. The monomer or building blocks of proteins are termed amino acids. Minerals that are required in relatively small quantities by humans. This will be the micronutrients. A mineral required in the synthesis of hemoglobin. This is iron. A vitamin needed to prevent poor night vision is vitamin A. And then organelles in the cytoplasm of the cell which plays a role in protein synthesis. This will be ribosome. A cell organelle concerned with the production of ATP during cellular respiration. This will be the mitochondrion of a cell. The upper lens of a microscope which magnifies an object is ocular lens or eyepiece. The membrane system in a cell where ribosomes occur, they occur on the endoplasmic reticulum. So now those are the biological terms, possible test and examination type of questions. And then the next question is question 1.3, where we have to indicate each of the diagrams in the column 1 applies to A only, B only, both A and B, or none in column 2. Now, question 1.3.1, there we have molecule A and molecule B, meaning they are bonded together or joined together. It means now there are two molecules. So in this answer here, it will be A only. 1.3.2, we have molecule A, and then there are three. We know now these are lipids, and in lipids we have fat and oils. So the answer here will be both A and B. Deficiency disease for a vitamin. Now, it does not indicate what type of vitamins. Now, we know beriberi and rickets, they are deficiency disorders that are caused by lack of vitamins. So, it will be both A and B. Nutrients needed in small quantities. This will be B only, micronutrients. Disorder caused by deficiency of iodine in the functioning of the thyroid gland is goiter, which is A only. The building blocks of nucleic acid are termed nucleotides. The answer there will be A only. Let's move to the next question. In this question here, we have a diagram that indicates a plant cell. Why we say it's a plant cell? One, it has a large vacuole, and if you see A there, it has a cell wall. And when we look at B there, it indicates a chloroplast. So when we study in this diagram there, it indicates that this type of a cell is a plant cell, and then the reasons are given there. It's because of a large vacuole, the presence of a cell wall, and the presence of a chloroplast. You look at the question there, is it a plant or animal cell? It is a plant cell. And then 3 observable reasons for the answer in question 2.1.1. The reasons are already given there. It has a large vacuole, the cell wall is present, and also the chloroplast is present there. Identify A to G, which means A to F, because we don't have G in the diagram. So our A will be the cell wall, and then B is the chloroplast, C will be the nucleus, the D is the cell membrane, E is the vacuole, and lastly F, it is the cytoplasm. State the names of 2 organic substances that are the main component of D, which is the cell membrane. It is the glycerol, and fatty acid. One function of B. Now if you look at B there, it is the chloroplast, which is important in photosynthesis. And then 2 functions of E. If you look at E there, it is the vacuole, which stores water and stores metabolic wastes. Moving on. There's another type of a cell there, which is different from the cell that we have looked at. And the below cell, it indicates the animal cell. Why we say it's an animal cell? Because now there's no cell wall that is present. There is no chloroplast that is present, and we can see the shape is irregular. Now identify the structures A and C. Now the A is the rough endoplasmic reticulum. We can see the ribosomes are attached to the endoplasmic reticulum, which indicates this as the rough endoplasmic reticulum. And then C, it is the mitochondrion of a cell, which is important in the production of energy through cellular respiration. What is the function of E? Now, if you see, E is a very small vacuole, which is mainly to store a little bit of water if it's present, store waste in the cell, metabolic wastes. Identify the type of cell. We have already identified this cell as an animal cell, and the reasons there is cell wall, it is absent, and is no chloroplast. And the shape of this cell is irregular. So those are the reasons why we identify this type of cell as the animal cell. So any of those reasons, as this question only requires one visible reason. Going to the next question there, complete the table, write your answer next to the question number of your answer book. Now we have an organelle of the cell, the function, and whether we find it in plant or animals or in both. So in question 2.2.1, we have Golgi apparatus, which are important in transportation or transporting proteins. And in question 2.2, we find them in both cells. In 2.3, we have to identify the cell organelle as we are given the function in cellular respiration, and we find it in both animals and cells. This is the mitochondrion. In question 2.2.4, we are not given the function, but we are given the name of the cell organelle, and it is only present in plant cells. It is photosynthesis. You know now the plant cells will use the chloroplast to produce their own food using light, which is photosynthesis. And then 2.2.5, it says gives a rigid shape to the cell, and it is only found in plant cell, and this will be the cell wall. Now this type of question requires you as a learner to know the names of the cell organelles that we find in the cell, their functions, or whether we find them in plant cells only, or in animal cells only, or in both. We'll just quickly go through again questions that are based on plant and animal cells. It says give labels for X, Y, Z. If you look at X, it is the nucleus of the cell. And then Y, the cell membrane. And then Z is the cytoplasm of the cell. Give the letter and the name of the part that stores water in plant. Now if you look there, that's as our plant cell, which is A, which is the vacuole. It is selectively permeable, which is Y. It's the cell membrane. It is the site of photosynthesis. We go to the plant cell because only plant cell has the chloroplast for photosynthesis, which is H. Responsible for cellular respiration, we know the mitochondrion of the cell is responsible for cellular respiration, which is I, mitochondrion. It is the site of photosynthesis, which are the ribosomes. If you look closely there, E serves as the ribosomes, as C is pointing at the half endoplasmic reticulum. Therefore, our answer there is E, which is the ribosome. Tabulate 3 differences between plant and animal cells. it is very important that you tabulate, as this question does indicate that you have to tabulate. As for table, you get 1 mark. Plant cells and animal cells. So basically, for providing the table, you get 1 mark for doing that. And then plant cells, we know that they have chloroplast. They have chloroplast. Animal cells, where chloroplast is absent, do not have chloroplast. Plant cells, they have a fixed shape, while animal cells, they have an irregular shape. Plant cells, the cell wall is present, while in animal cells, cell wall is absent. So for 7 marks, you indicate 3 differences and then also tabulate there. Can also include plant cells have large vacuole, and then in animal cells, small or absent vacuole. So when this type of question is marked, the differences are marked. They have to be differences. For example, say plant cell have chloroplast. Yes, they have chloroplast, is 1 mark. And then animal cells do not have chloroplast, is 1 mark. But if it is indicated like plant cells have chloroplast and then say animal cells have irregular shape, these are not differences. So therefore, the learner will forfeit the mark there. So it has to be the differences that are given. The next question is on enzyme activity. We know that enzymes are biological catalysts which speed up reactions that occur in our bodies. So if we quickly label 1, 4, and 5, we say now 1 is the substrate, 4 it is the enzyme substrate complex, and then 5 will be the products that are formed. What is the function of the part labeled number 2? You know, this is the enzyme, which is the biological catalyst. Speed up reactions. Moving on to the next question there, we have 2 graphs that indicates the composition of diets of 2 people, person A and person B. Now we calculate the difference in percentage of animal protein between the diets of person A and B. Now the person A, the percentage in animal protein there, it is 30. The difference, meaning we subtract, and then B, which is 5, and then 30 minus 5 will be 25%. Name one component of diets A and B which shows greater similarity of percentages. If you look at fat there, it has 10% in person A and then fat 10% in B. So it will be fat as the answer there. Say the ratio as a ratio the difference in percentages of cereal and sugar in person in diet A. Now sugar in diet A or cereal is 40 is to it's 10 there. Now 10 into 10 is 1 and 10 into 40 is 4. So the correct answer there will be 4 is to 1. Now if you look at the next question, there is an investigation type of a question where now was carried out to study the effect of temperature on the action of pepsin, which is an enzyme, a protein digesting enzyme present in the stomach fluid. The investigation was set up as follows. 10 grams of protein were placed in 5 test tubes. 20 mils of stomach fluid were added to each 5 test tubes, and the test tubes were taken and kept at different temperatures. After 24 hours, the content of each test tube were tested to determine the amount of protein that was digested. Now we determine the independent variable there. It is the variable that keeps on changing in the investigation, that is manipulated. So temperature is the variable that was changed in the investigation. Then the dependent variable, it is the amount of protein digested. State one way in which reliability of the investigation could be improved. Now there are two ways we can increase the reliability of an investigation. Repeat the investigation several times. Several times, not test, several times, and then you can also increase the sample size of the investigation. Now instead of maybe using 5 test tubes, you can use 10 or 15 or 20, increasing the sample size in the investigation. And now the question 3.2.3 requires the line graph to be drawn for the investigation. We have temperature as our independent variable, and we have the amount of digested protein as our independent variable. So when drawing a graph, we know that the independent variable, we place the independent variable on the x-axis and the dependent variable on the y-axis. And remember, what is important when drawing a graph is that your graph must have a title and you must place the variables on the correct axis. So temperature there, it is given in degrees Celsius, very important to indicate the unit, and then the amount of protein digested is given in grams, and therefore we must also provide an appropriate scaling for your axis. Now for temperature, you can use a scale of 10, 20, 30, 40, 50, 60, it goes to 70, this one. And then the amount protein there digested can be, 1, 2, 3, 4, 5, 6, 7, 8, 9. Remember, title or caption is very important to indicate. And then your graph is a line graph. Make sure that you draw the correct line graph. And then you just plot from there. Temperature of 5, and then amount digested is 0.5, which is half of 1. And then It's supposed to be somewhere there, and so on. So make sure you draw the correct type of graph. It's a line graph. You just put the dot on the correct coordinates, join the lines using your ruler, and then draw the correct line graph. State the possible reasons why no proteins digested at 70. We know that proteins they are sensitive to temperature, meaning that they have lost their structure and functioning, meaning that the proteins denatured at extreme high temperatures. Now, the last question we're going to look at is on an investigation that was done. This was an investigation. 3 test tubes were set up and illustrated in the diagram below. And then the test tubes were incubated at 37 degrees Celsius, which is the normal body temperature of humans, for 1 hour. The contents of each tube were tested by red or Milan's reagent respectively. The results are recorded in the table below. Note that the enzyme urease breaks down urea to ammonia. In the presence of ammonia can be detected by its effect on moist litmus paper. Now, which substance is present in plasma? We know that plasma contains cells, body cells, body cells, especially the white blood cells. White blood cells. Why were the test tube incubated at 37 degrees Celsius? Because 37 degrees Celsius is the normal body temperature of humans. Explain the purpose of test tube C in the investigation. Now, when you look at test tube C, only distilled water was put in and distilled in test tube C. Now, test tube C served as a control, and control, we use it to compare the results of the control together with the results that we find in investigation group. There are two ways in which reliability could be improved. We know that when you improve reliability, you repeat investigation several times, or you increase the sample size. Now, that question brings us to the end of this session, and I hope I was helpful in preparing you for the next upcoming end of the year examination. And I want to wish you best of luck in preparation of your examination. Rest well, take enough breaks, study very hard, and go to the examination prepared. Remember, the pandemic is still out there. Keep social distancing, sanitize, and wear your mask at all times, and keep safe. I want to wish you all the best in the final examination for life sciences. Remember to study very hard. From me, Mr. Ramkunupi, I would like to thank you for joining me in this session in preparation of your final test or examination paper. Thank you.